Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El-Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University. The Almighty God said in His last holy book to the humans, Al-Quran, in Surah Al-Ghashiyah, verse number 17, do you not look at the camels how they are created? قال الله العظيم في كتابه المقدس الأخير إلى البشر جميعا القرآن الكريم في سورة الغاشية الآية رقم 17 أفلا ينظرون إلى الإبل كيف خلقت. Before explanation and discussion the viral diseases of camels, we have to introduce the camels as animals. Camels are ancient, domesticated animals all over the world. They are the important source in desert areas for milk, meat, wool, transportation, and labor. Family Camelidae have different species. The New World humble camels are llama, alpaca, guanaca, vulcana species. The last three disappeared, and the only present one is llama, that present in North and South of America, as well as Australia. The second species of family Camelidae are humped old world camels. The one-humped camels is Camelus dromedarus or dromedary camel. Two-humped camel, Camelus bacterianus or bacterian camels. This picture showed the migration map of the historical Camelidae family. The current distribution of dromedaries and bacterian camels is presented in red and green colors. The last refugee of the wild two-humped camels in China and Mongolia are shown as dark green patches. This map was adapted from Misa Schumacher Armaco World. This figure showing the migration map of the historical Camelidae family. The current distribution of dromedaries and bacterian camels is presented in red and green colors. The last refugee of the world, two-humped camels in China and Mongolia, are shown as dark green patches. This map was adapted from Misa Schumacher Aramaco World. This is a collective slide showing dromedary camel diseases of OIE priorities. The bacterial diseases are brucellosis by Brucella abortus and Brucella melitensis, tuberculosis, paratuberculosis, pasteurellosis, anthrax, caseous lymphadenitis. The viral diseases are Camelpox, rabies, MERS, COVID-2, Rift Valley fever, African horse sickness, and blue tongue. The parasitic diseases are, most important one is trypanosomiasis. We will speak now about the viral diseases of camels. Management of diseases outbreaks in livestock farms. Viral diseases of camels. Camel pox disease is the most important viral disease affect family Camelidae. Camelpox is the only species-specific camel disease included in the OIE list of reportable diseases. Camelpox is a highly contagious skin disease and the most frequent infectious viral disease of the Old World camelids, dromedary and bacterian camels, that occurs in almost every country in which camel husbandry is practiced except Australia. Camelpox disease was initially described in Punjab of India in 1909, and later sporadic or widespread outbreaks have been reported, causing economic impact through loss of production and deaths. Pathogenic risk factor: Camelpox virus, an enveloped DNA virus which belongs to the genus Orthopox of the subfamily Poxviridae in the family Poxviridae. The picture of this virus is illustrated in the showing figure. This figure showing a cut surface of the pox virus under electron microscope after coloration. This figure showing cut away structure of a pox virus with example of vacina. The size of particle here is 360 multiplied by 270 multiplied by 250 nanometer. The envelope composed of three structures: surface tubule, outer membrane, and then the inner membrane. Then coming down to the lateral body, and then more deep in nucleoprotein that include the DNA structure of the virus, and then the core envelope which surrounded the nucleoprotein composed of core membrane and placidae layer. This figure shown more detailed based on the genetic formation of the virus, where in the upper part of the figure there is the outer membrane, inner membrane, core wall, core, DNA, genome, and viral enzyme. The lower part of the figure showing terminal loops in both composed of 7 base pair up to 54 base pair tandem repeats, and then at the 100 base pair we found the internal structure protein of inner membrane of the virus that composed of large subunit of messenger RNA capping enzyme and the viral DNA glycosylase and the internal structure of protein of an inner membrane of the virus. Then the subunit of early transcription protein, and then the subunit of RNA polymerase with membrane of protein of inner membrane of the virus. Small subunit of messenger RNA capping enzyme composed of 3 parts: nucleosidase, triphosphate phospholate, and the core protein of inner membrane of the virus. This genetic is an important in transcription, and the virus can replicate itself inside the cell of the host. Camelpox virions are large, enveloped, brick-shaped, and range in size from 265 up to 295 nanometers. The virion consists of an envelope, outer membrane covered with irregularly arranged tubular protein, 2 lateral bodies, and a core. The virus genome is double-stranded linear DNA of approximately 205 kilobase pair with covalently linked ends. In general, pox virions show high environmental stability and can remain infectious over several months, especially in crusts, serum, blood, and other animal excretions. Camel pox virus is sensitive to acidic media, alkaline media, boiling and autoclaving, ultraviolet rays ranged for about 245 nanometers wavelength. Camelpox virus is susceptible to various disinfectants. We shown here 5 of them. 0.5% of quaternary ammonium compound, 1% of peracetic acid, 1% of sodium hypochlorite, 1% of sodium hydroxide, 0.5 to 1% of formalin. In the following figure, we will discuss the geographical distribution of Camelpox disease all over the world. This picture of the world map showed countries report occurrence of pox outbreak by red color. They are most of African and East countries of Asia. According to what's illustrated and announced by OIE at 2020, this is the diagram of distribution of reported outbreaks of the most important viral diseases of camels in the sum of the world. Camelpox here is the firstly recorded disease, followed by contagious eczema or CPD, then the rabies, and ended by papillomatosis. For Camelpox disease susceptibility, young camels ranged between 1 to 3 years old are mostly susceptible. The recovered camels after infection by Camelpox had a long life immunity against this disease. Source of infection by viral Camelpox. The infection occurs through infected camels or skin scales that come from these infected camels and remain for long time in the soil. Mode of transmission of viral Camelpox disease. Number 1, introduction of new animals infected or in the incubatory period from contaminated areas or herds. Number 2, direct skin contact between sound and diseased animal. Number 3, inhalation of the virus from sick animals through ruptured vesicles or scars. Number 3, indirect transmission occurs via a contaminated environment such as grooming utensils or the same soil. Number 5, few days after co-watering with infected herds. We will speak here about effect. The incubation period of Camelpox mainly between 7 to 14 days post exposure to the infection. Sometimes it takes only 3 days with high dose of the virus of infection or during epizootics. In the followings, we will speak and declare the pathogenesis of Camelpox disease. Initial multiplication of Camelpox disease reported at the site of entry in both local and systemic infection. Camelpox virus will enter through portal of entry as direct skin contact or through inhalation of the virus at a systemic form, where viral multiplication nodes it at the nearby draining lymph nodes. This will cause in the future enlargement of the of these lymph nodes. Then after 7 to 14 days as incubation period, primary viremia will occur, and this clinically shown as feverish camel. Feverish camel will make himself toward the sun continuously, like sunflower plant, or seeking for cold water areas to be in. Then viral multiplication will occur in different tissues. In case of malignant form in youngs, the viral multiplication will occur in lungs, liver, and the digestive tract. This will lead to their death. While in the normal form of this disease, the viral multiplication will occur as cutaneous lesions post the secondary viremia, where cutaneous lesions appear as erythema, papule, vesicle, pustule, and crusts on face, neck, and under the tail. The viral multiplication occurs also in the uterus, but 87% of abortion of pregnant camels will occur due to the viremia. We will give you here a tip. Clinical picture of affected animals may develop localized or generalized pox lesion of varying severity depending on the age or the strain of Camelpox virus, as evidence suggests circulation of both mild and virulent strains. Clinical picture of Camelpox disease. Number 1. Fever and enlarged of lymph nodes. Number 2. Skin lesions reported post-fever by 2 to 3 days as erythema, papules, vesicles, pustules, and ended by scars. Number 3. The course of the disease is about 4 to 6 weeks. Number 4. Presence of 3 forms under field observation as the following. These 3 clinical forms are mild localized form without fever, severe generalized form with high fever, enlargement of lymph nodes and lesions on internal organs. Number 3, sporadic cases in adult animals, sometimes in pattern of persistent infection. The rest of clinical picture. Number 5, the lesions are commonly seen on head, including the lips, nostrils, ears, and eyelids, neck, perineum, and under the tail. In some cases, the mucous membrane of the oral cavity, limbs, genitalia, mammary glands are involved also. Number 7. Enlarged lymph nodes can be seen by naked eye, and some cases are accompanied by lacrimation and pendulous lower lip. Number 8. In severe cases, the virus can reach most of the internal organs, including the liver, kidneys, and the lungs, resulting in coughing and death of the affected animal, and this form is the malignant form of the camel pox disease. Number 9. Abortion of pregnant animals is caused by high fever or septicemia caused by secondary bacterial infection. This figure showing dromedary camels affected with camel pox showing pox lesion on lips and nostrils. This figure includes 2 pictures. The pictures with number A showing nasal discharges. The figure with number B showing severe skin lesions on head and face of an infected dromedary camel. Figure number 3 showing A camel showed papule and vesicles formation, edema of head and face conjunctivitis, with enlargement of mandibular lymph node affected with camel pox, showing pox lesions on lips and nostrils. Figure number 4 showing an infected dromedary camel with conjunctivitis. This figure explains a 10-month camel showing generalized vesicles and papules at areas of abdomen, chest, and inner thighs. Figure number 6 showing 4-year-old male camel with a history of fever, followed by scars, blindness within a week, and different stages of pox lesion. Figure number 7 showing a dromedary camel with different stages of pox lesion on face, fore and hind limbs, as well as under the tail. Figure number 8 showing dromedary camel with different stages of pox lesions on the skin of inner thigh and leg. This figure showing dromedary camel with different stages of pox lesions on mouth, lips, and nostrils, with a picture of corneal opacity. Figure number 10 showing a camel with different stages of pox lesions inside the mouth cavity, inner lips, and at the hard palate. This figure showing dromedary camel with different stages of pox lesions on the upper and lower lips and around the nostrils, with nasal and ocular discharge. Figure 12 showing a camel with different stages of pox lesions at upper and lower lips as well as around his nostrils. A dromedary camel with different stages of pox lesions at upper and lower lips from inside and outside with conjunctivitis. Figure 14 showing a camel with different stages of pox lesions at neck, face, and forelimbs. This figure showing a dromedary camel with different stages of pox lesions at neck and hind limbs. This picture of pox lesions on the perineum of a severely infected 3-year-old camel that results in its death. We will give you just a spot about the zoonotic importance of Camelpox disease. These 6 pictures showing the hand of an owner of an infected camel with Camelpox disease post 15 days of his infection had scattered scars, pock lesions at hands and fingers. Post-mortem pictures of a camel dead due to infection by Camelpox disease. These 5 figures showing a dead camel with different stages of pox lesions, where the pox lesions scattered on the tongue, lungs, liver, and abomasum of this dead camel. Camel's death usually reported in youngs due to the report of malignant form where the pox lesions had been found internally at trachea, intestine, lungs, as well as liver. A portion of pregnant camels usually reported during feverish time in about 87% of pregnant infected animals due to secondary bacterial infection. Laboratory diagnosis of pox disease in camel. Sampling. Samples from live camels are blood, double-paired serum, biopsy of skin lesions and scabs. Number 2, samples from dead camels are the trachea, lungs, liver, and intestine. The first step of laboratory diagnosis of Camelpox disease is the isolation of the virus on embryonated egg for detection of pox lesions, or as these figures shown tissue cultures of virus cells that show the cytopathic effect of Camelpox virus. Laboratory diagnosis of Camelpox disease also includes ELISA tests, detection of the virus under electron microscope, histopathological examination of skin lesions of deceased camels that displays cytoplasmic, swelling, vacuolation, ballooning of the keratinocytes of the outer stratum spinosum, acanthosis, and dermal layer infiltrated with inflammatory cells. The most accurate test used for diagnosis of Camelpox disease in its sensitivity and specificity is PCR test, which is a fast and sensitive method for the detection of Camelpox DNA. The PCR assays available to identify Camelpox virus are based on the detection of sequence encoding. DNA can be extracted from clinical material using numerous commercial kits. Here, the positive results were 243 base pair band appear at the electrophoresis gel. This picture showing the genetic composition of the Camelpox virus with phylogenetic tree constructed based on the gene encoding the A-type inclusion protein ATIP by UPGMA method in CLC main workbench 5.5 with 1000 bootstrap replicates. The number of each branch represent bootstrap values. The bar represents the genetic distance. Treatment of camels infected by Camelpox virus will divide into main treatment and supportive treatment. The main treatment including treatment of affected animals with antiviral medications. However, cost is high. The most common medication used to treat Camelpox is Cidofovir, a broad-spectrum antiviral that acts by inhibiting the viral DNA polymerase. Cidofovir has proven to be 100% effective for preventing deaths in infected camels. The supportive treatment of camels infected with Camelpox virus is by giving systemic antibiotics such as 10 or 20% of oxytetracycline and spraying affected parts with antiseptics such as potassium permanganate or other antibiotics is useful in preventing the ulcers from becoming infected by secondary bacterial infection. Traditional treatment of Camelpox disease in the field, nomads used liquid tar, ground seed of bitter apple, camel milk or mixture of sesame and table salt to rub affected areas. Treatment protocol also including giving antibiotics, antihistaminics and multivitamins for reducing the effects of the infection. Prevention and control of Camelpox disease. Programs to prevent and control of Camelpox disease by the veterinary authorities should include the following steps. Number 1. Sanitary measures. Number 2. Quarantine application on infected areas by isolation of infected camels and application of quarantine measures on sound 1, as well as restriction of camel movements. Number 3. Management of clean drinking water. Number 4. Avoidance of skin abrasions. Number 5. Vaccination of healthy camel in healthy non-infected herds. Vaccination in the face of an outbreak can be considered. Animals can be vaccinated by either live attenuated or inactivated vaccine. The live attenuated vaccine gives long-term protection against camelpox. However, a booster vaccination is recommended for young animals at the age of 8 to 12 months, 2 to 3 months after the initial vaccination, to avoid interference with maternal antibodies. According to OIE, camelpox is antifiable disease. We're keeping you on the right track, so please subscribe the channel to reach to the most newest knowledge about the epidemiology and different infectious diseases of different species of animals. See you later.